and welcome to this episode of OncLive Lung Cancer Talk. I'm your host, Dr. Josh Baumel from the Perelman School of Medicine at the University of Pennsylvania in Philadelphia. Joining me today in this discussion are my colleagues, all experts in the field of thoracic oncology. Dr. Hossein Borgai from the Fox Chase Cancer Center at Temple Health, also in Philadelphia. Dr. Balazs Halmos from the Montefiore Einstein Center for Cancer Care in New York. And Dr. Jarushka Naidu from the Johns Hopkins Kimmel Cancer Center in Baltimore. Today, we're going to talk about metastatic lung cancer, including a number of exciting new available therapies and results of key data sets presented at the ASCO 2020 virtual meeting. We'll talk about the impact on clinical decision-making and treatment selection for your patients. Let's get started on our first topic. So, Dr. Halmos, there has been a lot of talk recently about RET positive lung cancer. Can you talk to us a little bit about what the updated data are and where we are now?
Certainly. This has been a big year for RET, very specifically. Of course, the previous year was a big year for NTRK and a bunch of others, previously. So this year we've just learned about another new molecularly targetable subset of lung cancers with RET translocations. And let me just quickly remind us that RET is an important oncogene in a number of cancer types, including, of course, medullary thyroid cancers, where RET mutations occur frequently, up to 90% in familiar cases, such as in the MEN syndrome, 60% in sporadic cases. And you can see RET translocations in another type of thyroid cancer, papillary thyroid cancer, in about 10 to 20%. The same types of translocations occur in a small fraction, but a very important fraction of non-small cell lung cancers as well, about 1 to 2%. And this is very similar to ALK and ROS in terms of, these are fusion proteins that get generated. The fusion protein basically merges a protein with the dimerization function with the tyrosine kinase portion of RET, and as a result, it becomes oncogenic. But RET kinase has been actionable for quite a few years, though, through the availability of these multi-targeted kinase inhibitors such as vandatanib, et cetera. So we've known about some actionability, but these drugs have been very toxic in a way and not very specific. So they really haven't gained that much traction. So this year was really a beautiful breakthrough year now with the development of very specific and potent RET inhibitors, actually two of them that are leading the pack at the moment. And one that we've heard about even last year, selpercatinib, has shown really dramatic responses in patients with RET translocation positive lung cancers, both in the treatment refractory, but also in the treatment naive setting, to the point that when you look at the waterfall plots, basically every single person seems to benefit to some extent, with response rates in the 60% or so range in treatment refractory in terms of platinum refractory patients, and up to 80% in treatment naive patients. Also, not just that these medications are very effective, both selpercatinib and the second drug, pralsetinib, but the RET kinase is not very important for the adult human being, fortunately. As a result, the toxicity is not all that much. In fact, no major dose-limiting toxicities have been noted with these compounds. These are very safe, very potent. Selpercatinib now has been approved. It's got the commercial name called Retevmo, and RET, of course, comes from RET. Do you guys know what the MO is coming from?
No, I think there's a mystical creature that decides these names. I think that there's a riddle that you have to answer. I'm not sure how that works.
Mo comes from mojo. It's got a lot of mojo. That's where it's coming from.
The mojo. Speaking of, we're going to talk a little bit about some brand names shortly that some would wonder about. But in this case, Dr. Borgai, Dr. Naidu, are you currently testing for RET, and in which patients are you testing?
Yeah, I can jump in. So here at Hopkins, and I know at many large academic centers, we do widespread panel testing with NGS that includes testing for RET fusions. And I think many of us have been proponents of this approach because it allows us to conserve tissue while doing massive parallel sequencing. But obviously, this is a different type of alteration, and those who may be more used to testing for individual alterations that were associated with improved therapies or approved therapies now would have to change or alter some of their approaches in order to test for a RET fusion. So I think that's a very important take-home message.
Mm-hmm.
Yeah, thanks, Josh. Similar to what you just heard, we have also been testing for the RET fusions. We actually have a 500 gene panel fusion that basically gets done on all the tissue that we submit. And as far as who we test, I think just like we advocate for everybody with a diagnosis of non-small cell lung cancer, particularly non-squamous, to be tested with a NGS broad spectrum panel, we're doing the same with all of these fusions for everybody who comes in with a diagnosis of advanced non-squamous non-small cell lung cancer. Occasionally, patients who are diagnosed with squamous cell histology, particularly if they're never smokers, we do test them. And the goal here, at least for us, is to expand the testing to everyone, regardless of histology with non-small cell lung cancer.
And when you guys are doing these testing, and Dr. Halmos, you can jump in here too. Is it a DNA-based assay? Is it an RNA-based assay? Are you using both? How are you incorporating these tests?
Let me just comment on for RET, you can use a couple of different platforms. FISH is actually acceptable for RET fusion detection as well, PCR too. But in reality, since we do this as part of a panel, what ultimately comes down to is the best test tends to be NGS-based platforms. I have to highlight that ctDNA testing is actually pretty good, for these as well. And actually, the Blueprint Arrow studies, have shown a lot of interesting ctDNA data in terms of ctDNA clearance as well, pralsetinib. So that's definitely an appropriate platform to use in the usual settings where ctDNA we find appropriate in our lung cancer patients. And one thing that I failed to mention, because I got stuck with the mojo situation, is that, of course, CNS penetration is very important, for all of these TKIs. And both salpercatinib and pralsetinib seem to have, pretty good CNS penetration and CNS activity, as we've seen from, the early but important sets of data.
And so if you guys have a patient with RET fusion positive disease, you identify it on initial diagnosis, what line are you using it? Are you using it before other treatments or using it after?
I think certainly if I saw someone with a RET fusion, I would think about giving this first line. If I felt that this was, a patient who could wait for some of this testing to come back. I think a lot of the time it's the wait time for the testing to come back, which is usually 2 to 3 weeks, that makes a decision for us. The burden of the disease, the symptomatology of the patient. But certainly I'm a big proponent of waiting for the full molecular panel to come back. And now that panel includes RET fusions.
Okay, I agree. I think I would rather use it in the front line also, again, given the right clinical scenario. And as it was just mentioned, if the patient can wait for the results of the testing, I think it's completely appropriate to offer it frontline.
I totally agree. And let me just highlight that the activity of these compounds is very similarly in range with EGFR, ALK, and RAS inhibitors, where we've accepted frontline use of the targeted agent. So given the excellent activity and also limited toxicity, there's no particular reason not to think that we could follow the same guidelines. And that's why it seemed so upsetting for all of us last year that there's a randomized study being planned, comparing, chemoimmunotherapy versus targeted compound in this particular setting, where it just makes really not a whole lot of sense based on all that we know.
Yeah, it's interesting. These drugs, we keep doing the same thing over again, right? So we have a highly active TKI, we compare it to chemo, and shockingly, it works better. And then so we're like, oh, we have another highly active TKI. Let me compare this to chemotherapy. Let's see what happens. And if you actually pull up a table of all the EGFR TKIs versus chemo, it gets enormously overwhelming how redundant these trials are. And there are reasons why they've done it in terms of agency regulations, but I think that we as a scientific community, I completely agree, need to come out and say, look, these drugs are better. They're better tolerated. They have better activity. And I completely agree they should be used in the first line.
So where do you think the lesion is? Is it really from a regulatory agency? Because when we talk to our European colleagues, they actually tell us that to get something approved, even in a patient population that has a specific target, you need to have a randomized study versus standard of care, which I think, again, just like you and Balazs just said, it just doesn't make a lot of sense to most of us. But we don't know how to overcome that regulatory block and not have to do these randomized studies.
I think that the barrier here is that these are very expensive drugs. There's big numbers associated with this, big cost. And I think regulatory agencies don't tend to change terribly quickly, but I think it's very appropriate for us to consider moving towards changing this, because I wouldn't randomize a patient to chemo, IO versus selpercatinib or pralsetinib here.
But at the same time, to play the devil's advocate, do we know for sure? I guess there's always that element that, you know, in a clinical practice, when you're dealing with a complex patient, do we know for sure that it is better for the frontline management than, let's say, chemo IO in this case, because that's what seems to be the study. So I think there's always this question as to whether a randomized study would still play a role in truly defining the role that these drugs can play, at least in the frontline setting.